Now we are going to see the application of genetically modified organisms, competent plants in agriculture, and with that we are going to understand about this. Now what is this? This is RNAi. Okay. This is RNA interference. I stands for interference. Now it is such an important process that two scientists who study about this, Andrew Fire and Craig Mallow, they got Nobel Prize for RNA interference in 2006. That is quite recent. Okay. So what was this RNA interference? It has its application in agriculture as well. We study this RNA interference in terms of a nematode known as Melidiogyne incognitia, which is a nematode, as I told you, incognitia, that is responsible for infecting the roots of tobacco plant. Now you know tobacco has a pretty economic importance because it makes the smoking world jubilant. It is responsible for making cigarettes, so it has an economic importance, though that causes a lot of cases of lung cancer. But this particular nematode was infecting the roots of tobacco plant. Now over here, in order to control that infestation by this particular organism, the mechanism of RNA interference was brought into use. What is this RNA interference? As the name suggests, RNA will come and interfere with the causative gene that is affecting the tobacco plant. That means the gene which is responsible for causing harm to the tobacco plant present inside this particular nematode will be interfered by a double-stranded RNA. Okay. Now this RNA interference is observed in many eukaryotic cells. Why these eukaryotic cells have this mechanism of RNA interference, that is otherwise known as silencing? This acts as a self-defense mechanism where certain parts of the genome of the eukaryotes is providing defense, where double-stranded DNA is being produced and it silences the particular part of the genome, so that it acts as a defense mechanism. Okay. So we have a double-stranded DNA which has both sense and antisense strand. Okay. And they are complementary to each other, that is for sure. They make a double-stranded RNA. This double-stranded RNA jumps from one side to another, acts as a transposon. What are transposons? Transposons are jumping genes that they move from one point to another. It acts as transposon and helps in the cellular defense. Okay. It is the part of cellular defense mechanism in certain eukaryotes. You just have to remember that it was observed in eukaryotes, showing the mechanism of silencing, which is an immune mechanism of eukaryotes, where the jumping genes, or otherwise known as transposons, that means from one site to another. The gene is almost similar, it goes from one side to another. That is not the case, but the gene is similar, it produces similar RNA. So for that we have double-stranded RNA for silencing the act of a particular gene. Now what happens in this mechanism where we use it for getting plants which are capable of avoiding the infection? See, a gene for the sense strand and a gene for the antisense strand would be incorporated into the plasmid. Okay, that plasmid would be put into agrobacterium. Okay, the agrobacterium would go and infect the tobacco plant. The tobacco plant then will have gene 1 and gene 2 copied inside it. These genes would produce sense strand and antisense strand. Both sense strand and antisense strand would come together. Okay. Then they would go into the genome, go near the genome. When the genome codes for genome of this particular enzyme or this particular nematode, it codes for a specific gene. The RNA that is present because of incorporation of these genes would go and silence that gene of nematode. Okay. I am going to repeat it so that you get an idea. Nematode has a specific gene which is causing problem for the plant. Now, what you want to do is you want to create such a plant which does not allow the gene of this nematode to express itself. So, you have to do silencing of that gene. How do you do silencing of that gene? You use double-stranded RNA that can bring about silencing of that particular gene which belongs to this nematode. When the gene is inside the tobacco plant, now the tobacco plant has production of double-stranded RNA inside it. That double-stranded RNA would go and curtail or silence the infectious gene of Melioidogyne incognitia, and it will not be able to survive within the tobacco plant. And you see, thus we use RNA interference for our own usage, where the tobacco plants are no more going to get infected by this particular nematode. Why? Because they have double-stranded RNA being produced inside them. Who produces it? It is the plasmid that was introduced by agrobacterium. Now, that plasmid has a recombinant DNA which has genes for production of both sense and antisense strand to produce double-stranded RNA that is going to silence the gene of this particular nematode. So, this is what RNA interference is all about, that you have to keep in your mind, and the example of this particular nematode would help you in understanding. You do not have to go beyond it, because there are many concepts like dimer concept, how they change into small nucleotides, how the sense and antisense strand get separated. Do not get into depth of that. Just remember that the antisense and sense would come together, two different genes would be coding for them, they would come together, they would form double-stranded RNA. That double-stranded RNA is going to do silencing at transcriptional level. Now, silencing of what? The gene which is responsible for pathogenic activity of this particular nematode. Okay. So, this is all about RNA interference that you have to remember.